So now I'd like to welcome up our panelists for the first session, focusing on aquatic habitat research. Our panel includes Dylan Yakich, Andrew Wooley, Kelly Chase, and Matthew Bunting. If you'd like to go ahead and come up as I read out your introduction. Dylan Yakich is a fisheries biologist with the Florida Fish and Wildlife Conservation Commission's Fisheries Independent Monitoring Program. She is originally from the suburbs of Chicago and has been passionate about marine biology since she started diving at a young age. Dylan obtained a BS in biology with a focus on organismal and ecological biology from Florida Gulf Coast University. She worked as a laboratory assistant in the invertebrate zoology laboratory and volunteered with FWC as an undergraduate. Dylan has spent the last 3 years as an OPS Biological Scientist 3 with FWC, conducting research on the small-tooth sawfish. Andrew Wooley is a fisheries biologist with the Florida Fish and Wildlife Conservation Commission at their Charlotte Harbor Field Lab. He was born and raised in Southwest Florida and has spent the last 7 years with FWC studying the fishes of Charlotte Harbor with a focus on small-tooth sawfish. In addition to his work for FWC, Andrew is studying the movement and habitat use of juvenile small-tooth sawfish within the harbor for his graduate research at the University of Florida. Kelly Chase is a fisheries biologist with the Florida Wildlife Research Institute's Fisheries Independent Monitoring Charlotte Harbor Field Lab, where he studies fish community dynamics and juvenile sport fish ecology. In 2022, Kelly began pursuing his MS in Fisheries and Aquatic Sciences at University of Florida, examining the effects of habitat shifts on fish communities and the feeding ecology of juvenile common snook. Matthew Bunting graduated from Florida Gulf Coast University with a bachelor's degree in marine science and from the University of Florida with a master's of science in fisheries and aquatic sciences. Currently works for the FWC and the Fisheries Independent Monitoring Program, and his work focuses on ontogenic habitat shifts in sport fish, coastal pond functions as nursery habitats, and coastal habitat restoration design and monitoring. Each is going to give a brief presentation on their respective topics, followed by audience Q&A. Please feel free to enter your questions again throughout this presentation, and at the end, our panelists will respond to as many as time allows. And with that, I'm going to turn it over to you, Dylan. Thank you.
Hello. Good morning. So I'll be giving a presentation on the effects of marine debris and human interactions on the small-tooth sawfish. So first I'll start with a brief background on small-tooth sawfish and then discuss their historical marine debris issues, get into their current marine debris issues, specifically with ball bungees, and then potential solutions and our hotline. Small-tooth sawfish are an endangered species of ray. Historically, they are found in coastal waters from Texas throughout the Gulf, around Florida and up to North Carolina. But unfortunately now the species is primarily only found in South Florida. In 1992, the species was protected from harvest, and as we all know, the Everglades National Park has been a protected area since the late 40s, which certainly sawfish were able to take advantage of this, and in 1995, gill nets were banned in Florida waters. This wasn't because of sawfish, but they certainly did benefit from it. And then in 2003 is when they're enlisted on the Endangered Species Act, and after enlisting, soon a recovery plan was implemented. So the sawfish population mainly declined due to unintentional overfishing by commercial fisheries, mainly caught as bycatch in gill nets. And the species still face threats due to bycatch and commercial fishing trawls, as well as habitat loss due to coastal development, and as you'll see throughout this presentation, marine debris. They typically have 22 to 30 teeth on each side of their rostrum. This is mainly used for feeding and defense. The pictures displayed here show how susceptible the rostrum is to an entanglement, this toothy rostrum. The photo on the right was a sawfish we caught in the Caloosahatchee River. She was found to have an entire cast net entangled in her rostrum. Once after workup and prior to release, we were able to successfully remove this off of her. So now getting into their historical marine debris issues, Sites and Palakis conducted a study from January 1999 to November 2005. This is really the beginning efforts of our angler guidelines and where our hotline originated from that we use today on a daily basis that helps our sampling, direct our sampling efforts. So this study was just over, a total of 3,000 encounter reports, and of that, 50 reported entangled, injured, or dead due to marine debris. The photos here, you can see this fish was caught in a cast net, and it was found to have a PVC pipe wrapped around the base of its rostrum. The bottom middle photo shows these distinctive notches at the front of its head, as well as half the teeth are missing or broken off that rostrum. In the top middle photo, you can see how much biofouling there is on it, suggesting that this has been on the fish for quite some time. This was another fish reported to us through the hotline that we did a necropsy on. Again, we see these similar notches at the base of its rostrum, as well as each teeth on either side of the rostrum are completely gone. But what was also particular about this fish was that it also had recreational fishing line looped through its head as well as coming out of its mouth. So not only did it have one marine debris issue, but several. Another fish we had caught during our sampling efforts found to have a bunch of recreational fishing line embedded into its rostrum. This is where our guidelines come into play, and we ask that people cut the line as close to the hook as possible, because at some point this line can become embedded as the fish continues to grow. And in severe cases like this one, the rostrum can be compromised and the fish is unable to use it to its full potential. The top photo is the same fish as the previous slide in comparison to what a healthy rostrum should look like. Historically, various forms of elastic bands have been found on sawfish, from hair ties to rubber bands. Once when this type of debris gets into the water, it sinks to the bottom, and as we know, sawfish are a bottom-dwelling species. So as they're swimming and this gets hooked on their rostrum, as they continue to move forward, it continues to pushes down because the fish do not swim backwards. It's really up to anglers' discretion if they feel comfortable and confident enough to remove any marine debris off fish. The bottom photos show other examples of why we suggest and recommend not to put any ropes on these fish. This was a hotline report in the 10,000 Islands, and this fish was found to have rope wrapped around the base of its rostrum as well as the base of its caudal tail. Unfortunately, it's not unusual for us to receive reports of sawfish that have rostrums removed or broken rostrums with various lengths of rostrum left. We know that sawfish are still targeted for rostrum removal, and as I mentioned earlier in this slide, they are still caught as bycatch in commercial shrimping trawls, which is the photo that you see on the right. We hope that these fish would be treated with better care, but unfortunately, they are still caught in a large juvenile to adult fish like this. When it's lifted in the back of the boat, this can cause additional weaknesses and breaks in their rostrum. So let's fast forward almost 2 decades. What are we seeing new? What are we seeing different? This current study was over the same time period as previously, and it was just about the same total encounter reports, but it was 4 times the amount of fish that were reported entangled, injured, or dead due to marine debris. So yes, unfortunately, we do still see all the same historical marine debris, if not more. including miscellaneous household items. Not only the photos you see here, but we've had hotline reports of sawfish entangled in sunglasses, swimming visors, plastic bags. This is when we began to start seeing something a little bit new, something we weren't as familiar with. These distinct notches right behind their spiracles, the severe damage wrapped around their head, and these black elastic bands that we were unsure of what they were or where they were coming from. Until one report in particular, this fish was accidentally caught in the Caloosahatchee River. It was found to have this black elastic band, and the angler was comfortable and confident enough to remove this. He had suggested that he thought it was an ankle bracelet to a boogie board, and he was kind enough to be able to send this in to us, and we were able to further examine it and take a look at it. And what we had realized, or what we hypothesized, was that this was a ball bungee with a piece of tarp on it. As we continued our sampling efforts and continued to receive more reports of fish, we were able to have full confidence that these were coming from boat lift canopy covers. And at least 20 sawfish have been entangled in these ball bungees reported to our hotline or sampling efforts. So the bottom photo, you can see if any piece of strap is left behind, it disconnects from the main part of the canopy, especially during tropical storm events. Unfortunately, it goes to show that these elastic bands do not break, and actually the tarp rips from itself. This is just one particular brand and style canopy. This location is in Cape Coral, Florida, and as we all know, Cape Coral has extensive mile-long canal systems found there, and pretty much every canal there has boat lift with boat lift canopy covers. This one cover alone has 66 ball bungees on it, and as a side note, after Hurricane Ian, only 10 of these ball bungees were recovered, one with a strap still on it. If any of you would like to see it, I do have one with me. So you can imagine how many of these ball bungees can get into the water. And so this was the first fish that we know of that died with a ball bungee around its gills. The top middle photo shows the actual gill arch of this fish being exposed once when this ball bungee was removed. We know that these are resilient fish and they aren't always dying, but in this case, it can definitely be severe and interfere with gill function. In 2021, 3 sawfish were caught by us with these distinct notches, including the one that you see here with the ball bungee still around its head. Once when we had removed this, the indents left behind were several inches deep, and as the fish was actively breathing, we could see water coming out of there. And to put it in perspective, this is an 8-foot fish. So in summary, this is an ongoing problem in the city of Cape Coral and Fort Myers. We really want to start getting the awareness out there for residents that live on the water there. But the results of this current study did show that hook and line gear are still the #1 common form of marine debris found on sawfish, with ball bungees taking a quick rising in a very short period of time. So potential solutions. Hopefully throughout this presentation, you've seen the precautions to take when it comes to your boat lift canopy maintenance, whether it's installing, removing, or replacing, especially if you have time to prepare ahead for hurricane events. At the bare minimum, we could suggest to cut the loops of these ball bungees. But we do have ongoing efforts with the city of Cape Coral. We were featured in their Canal Watch newsletter, which started to educate and inform the residents there, the outreach that they need to know, and hopefully better maintenance to their boat lift canopy cover. I would like to emphasize that this is not just a localized issue, but it is a statewide problem as well as in the Florida Keys. So you can see on your left, this is our outreach sign. You can find these at local bait shops, marinas, boat ramps, encouraging the public to report any sawfish encounters to us. And with that said, I'd like to thank NOAA and NIPS for funding this research, citizens that take the time to call in and report their sawfish encounters to us, and Charlotte Harbor Field Lab, who all help to contribute to these data collection. If any of you have any recommendations for further locations to put our outreach sign or would like to share any sawfish encounters, please find me or any one of us later on. Thank you.
All right, so I'm Andrew Wooley, and I'll be talking about my work with the ontogenetic shifts in habitat use by the endangered sawfish here in Charlotte Harbor. Dylan already covered a little bit of this, but I'll start off with a little bit of background information about sawfish and get into the work that we've been doing here since the early 2000s with sawfish in Charlotte Harbor. Then I'll talk about the research that I'm doing for my thesis and get into the methods and some of the preliminary results. So as Dylan just mentioned, sawfish are a critically endangered species of ray that we're lucky enough to have here in Charlotte Harbor. And throughout this talk, I'll be referring to a couple different size classes of sawfish. So I want to orient you to what I'll be referring to when I talk about these. So I'll start off with small juveniles. When I refer to small juvenile sawfish, I'm talking about both young of the year fish, so fish that were born this season. They're born at about 2.5 feet. And then they double in size in their first year. So a small juvenile is anything between 2.5 feet and maybe 5 or 6 feet. And then I'll also be referring to large juveniles, which are those fish that are maybe 2 years plus, and those would be 7 to 10 feet long. And just keep in mind that small juveniles are still potentially 6-foot fish, and large juveniles are 7 to 10 feet, but they're still juveniles. Anything above that would be considered an adult. So for my thesis, the purpose of the thesis was to identify ontogenetic shifts in habitat use related to sex and size, so looking at how they use their habitat differently as they grow and determining differences between the way that they do that between males and females. Also, we wanted to evaluate the residency of sawfish within Charlotte Harbor, so we want to look at not only how they're moving about the harbor, but how long they're staying there and what they're doing while they're here. Historically, as I mentioned, we started doing research on sawfish back in 2004, and from 2004 to 2017, we were mainly using gill nets in Charlotte Harbor, and these gill nets were mostly set just in the river systems up in the Peace River and the mouth of the Myakka, as well as the Caloosahatchee River. And these gill nets were mainly used to target just small juvenile fish, so those fish under about 6 feet. And these nets were set from the shoreline out perpendicularly. And when we caught small juveniles with these nets, we were really only able to put external V9 acoustic tags. So because they're external, they're very small. They had a very short-term battery life of only maybe 6 months to a year if we were super lucky. So we weren't really able to get much long-term data from these small fish. For this study, we were looking at 2017 to 2021, and in that time period, we set over 1,000 gill nets in Charlotte Harbor. And we use these gill nets in both our random sampling and directed sampling. Random sampling is used to determine relative abundance of sawfish. So we use a computer that spits out random coordinates in a generated universe. We go to those coordinates and we set nets regardless of whether or not we actively see a sawfish or not. Directed sampling is using our sawfish hotline like Dylan just mentioned, and we respond actively to reports, or we'll go out to areas where we've had historical reports where we know there might be sawfish, and we'll go and we'll actually look for sawfish, and if we see them, we'll try to catch them. As I mentioned, these gill nets are set from shore, extending out perpendicularly from the shoreline into slightly deeper water, but generally, because they're so close to shore, the average depth of these nets was only about 1/2 meter or knee deep. And then in 2017, we received permission to start internally tagging sawfish. So with these internal tags, we could put longer battery life tags inside them, and we just make an incision in the abdominal cavity, and we can put in 5 to 10 year acoustic tags. And we continued to use the gill nets, just like we have from 2004 to 2017. We still use those to this day, but we also implemented some new sampling gears to start targeting different size classes of fish. So now we use hook gear as well, which consists of drum lines and hook and line gear. Again, from 2017 to 2021, we did almost 1,000 hook gear sets as well. And we're using drum lines, which are essentially like a single hook long line attached to a weight on the bottom, and hook and line gear, meaning just basically rod and reel gear like you would use anytime you go fishing. And generally speaking, these gears were set in very deep water compared to the gill nets. So we use gill nets in shallow water, and we use hook and line gear or hook gear in deep water between usually about 10 to 12 feet and sometimes even deeper. If we combine these gears over the study period, in those 5 years, we captured 300 plus sawfish throughout the harbor. And you can see the majority of our sawfish are on this left-hand side. They're very small juveniles. They're less than 1.5 meters. But we have started to catch some larger fish over the years. And we've been identifying areas where we can catch large juveniles and even sprinkling in a few adults. You'll see up there around the 3 and 4 meter mark. So just to review what we kind of already know from previous work on sawfish here in Charlotte Harbor, we know that small juveniles occupy 2 distinct nurseries in the harbor, one up in the upper harbor between the mouth of the Mayaca and the Peace River, and then one down in the Caloosahatchee River. And within those 2 nurseries, these small juveniles occupy extremely shallow water, and they exhibit high sight fidelity. So wherever they're born, they'll just push up into as shallow water as possible, and they'll stay there for quite some time trying to avoid predators and just grow a little bit larger before they kind of get their wits about them and go into deeper water. We also know that large juveniles are pretty much found throughout the harbor in different pockets of deep water. And at some point, these sawfish reach a great enough size where they'll actually completely leave the harbor. And once they leave, they'll move about the entire state of Florida and join kind of a migratory population. So to figure out more about what sawfish are doing before they leave the harbor, we put out 180 internal tags from 2017 to 2021, most of which being 10-year internal tags. So we can essentially track these fish with acoustic data from the time that they're sometimes young of the year fish all the way potentially to adulthood. We also expanded our receiver coverage for our acoustic array in 2017 because, as I mentioned, our previous research was mainly focused on small juveniles in the river systems, so we had a lot of acoustic receivers there, but we wanted to put some in these areas outlined in the orange box in some deeper areas in the middle of the harbor where we would expect these small juveniles to transition to when they get larger, and we would expect large juveniles to be hanging out once we tag them. And this is kind of what instigated my particular project, was we were looking at the length frequencies, and just to orient you here, this is 2 length frequencies stacked on top of each other. So the red bars are fish that were caught in gill nets, the blue bars are fish that were caught on our hook gear, and the other third color there is just where they overlap. And there's pretty minimal overlap here, and we did find statistical differences between the size classes caught in hook gear and gill nets. So we noticed that, okay, well, we're catching mostly small fish in gill nets, we're catching mostly large fish on hook gear. And if we look at that a little bit differently, we see this kind of primary habitat shift, where if we plot the stretch total length of sawfish on the x-axis and the capture depth on the y-axis, if we look at this in quadrants, the bottom left-hand corner there, those are all small sawfish that were caught in very shallow water. The upper right quadrant are all large sawfish that were caught in deep water. And this suggests that based on our gear type and our sampling methods, we're catching small fish in shallow water, and they're transitioning to deeper water as they get bigger. There's a couple of exceptions here that I want to point out to you. These sawfish that are circled in the red circle there, those are all sawfish that we would consider smaller than we would expect for how deep the water was that they were caught in. And we believe this is because all of these sawfish were caught at the foot of bridge pilings throughout the harbor. So we think that these kind of intermediate sized sawfish are using this transitional habitat where they're going from the shallow shorelines, they find the bridge pilings, and they kind of use those pilings to get out into deeper water, but have some structure for protection before they get into these deep holes. The other one here that's kind of an outlier is this orange triangle down in the bottom right-hand corner. And unfortunately, this was a huge adult female sawfish that we caught in the Caloosahatchee River. We caught her in just above knee-deep water, and unfortunately someone had completely removed her rostrum, so we do not believe that she was behaving typically of what we would expect from a normal adult female of that size. So that was the first habitat shift into deeper water, and then we've also identified this potential secondary habitat shift, where we think that these sawfish at a certain size, they're moving away from the rivers, but they're staying in Charlotte Harbor. So there's this kind of range expansion or home range expansion, leaving the river systems after about 2 years and then going into the main parts of the harbor. But they don't just completely leave the rivers, they might bounce back and forth between the rivers and the harbor. And in some cases, they even completely switch nurseries. So if they were born in the Caloosahatchee River, they might hang out there for a little while, and then they'll actually go up into the Peace River and vice versa, which is something that we never really saw before when just analyzing movements from small juveniles. So to get more at this, we, as I mentioned, tagged a bunch of fish, and some of these fish, they remained in the estuary after being tagged for upwards of 3 or 4 years. And bear in mind, we're tagging these fish at all different sizes, so all different ages. So some of these fish were staying in the estuary much longer than we expected. Previous research suggested maybe 2 or 3 years. It's looking like perhaps 3 to 4 years, maybe even more. And of the sawfish that we tagged, over 100 of them actually stayed in the harbor for more than a year after being tagged. And we wanted to look at how those movements were taking place. So just to give you an example, we broke down our receiver array into these 4 sections. The dark blue circles here in the bottom, those are the Caloosahatchee River. Those are receiver stations within the river. And then the orange circles are going to be receiver stations outside of the river in San Carlos Bay and Lower Matlacha Pass. The blue X's, the light blue X's, those are going to be receivers in kind of the middle of the harbor off of Cape Hayes and the east wall. And then the upper harbor receivers are these purple diamonds. And we just took a quick look at 3 of the sawfish just to get an idea of what kind of movements they were making. These 3 sawfish were all tagged in the same place at the same size, but they were not necessarily tagged at the same time. So you can see fish 2 was tagged a year prior to the other 2 fish. And all of these fish, as I mentioned, they were tagged the same size. They were 1 year old approximately when we tagged them, and they stayed in that blue area, which is the Caloosahatchee River, for a year after they were tagged. You'll see a little blip on fish 3 where it goes out into the orange just right there at the beginning. We think that's just a receiver coverage issue. We think that it was at the mouth of the river and never actually left. It just got heard by a receiver outside. But all 3 of these fish, you can see they stayed in the river for the first year, and then all 3 of them moved out into those orange dots, those orange receivers, which was the mouth of the river and San Carlos Bay, kind of in the lower harbor. And fish number 1 and fish number 2 there at the bottom, they just kind of flop back and forth between the lower harbor out in San Carlos Bay and Matlacha Pass, and going back into the river, and just kind of moving to and from. Fish number 3, however, did something a little bit different. You can see that fish left the Caloosahatchee once it got to about 2 years old, went up through Matlacha Pass all the way up into the Peace River, and then started bouncing back and forth between the Peace River, the middle of the harbor, and Matlacha Pass, using a huge home range compared to the small juveniles that would just be in the rivers. One thing to note here too is that, as I mentioned, all 3 of these fish were tagged at approximately 1 year old, and all of these data sets last for 3 years. So these fish were all 4 years old at the end of this time period, roughly, and at the end of this time period, all 3 of these fish completely left the harbor. So we do believe that somewhere around 4 years is likely the time that these large juveniles are starting to move out of the harbor and migrate throughout the state. So in the future, we want to continue looking at these. We want to analyze the rest of the 100+ sawfish that we tagged, and we want to compare how these depth changes as well as the home range changes are taking place between animals of different size, animals of different sex, and potential seasonality and other effects that might be causing these changes. We also want to further investigate this residency of sawfish in the harbor, because as I mentioned, previous research suggested maybe it was only 2 or 3 years. But we want to know how long are sawfish staying in the harbor before they become migratory, and that'll give us a good idea of how many different potential age classes are in the harbor at any one time, to give us a better idea of how many sawfish are here and what they're doing. So thank you for watching, and I want to thank our funding sources at NOAA and NIMS and all of the folks at our Charlotte Harbor Field Lab for helping out with this research and answering all the phone calls on the sawfish hotline that Dylan mentioned. And I'll take any questions at the end.
Is it on? All right, cool. Get that clicker. Good morning, everybody. My name is Kelly Chase, and I'm going to be talking today a little bit about the effects of seagrass loss in some of our tidal creeks on fish communities inhabiting those systems. So tidal creeks are an important nursery habitat for a suite of fauna, including economically and ecologically important species such as tarpon and snook. They're pathways of energetic transfer, so all that production on the marsh surface can be funneled through these systems and into the open estuary. They participate in important biogeochemical processes as well and provide environmental aesthetics. So if you've ever explored some of these areas, you may have felt more so like you were in a Jurassic Park movie than necessarily the hustle and bustle of Southwest Florida. Considering their position on the landscape, however, as they traverse the land-water interface, they may be especially vulnerable to the effects of coastal development. Coral Creek, located on the Cape Hayes Peninsula here in Charlotte Harbor, is one of our larger tidal systems. It's got 3 distinct sections, lower, east, and west, and it's somewhat characteristically unique compared to the majority of the other tidal tributaries we see. So for instance, especially on that eastern portion, it lacks kind of that classical meandering morphology with point bars and cut banks, and is more so has a consistent shallow depth throughout, and this unique association with seagrass, which extends well into the system. As you can see from this image, the watershed has been extensively developed, and it might be kind of hard to see, but it's important to point out that on that western branch there's actually a weir just below that word west, and that's impeded historic sheet flow from that upper section of that branch. In the early 2000s, Coral Creek began experiencing these large filamentous green algae blooms. And over the years, we've learned that these algae blooms have recurred with varying frequency and intensity. And it's also important to point out that it appears that the eastern portion of Coral Creek has really experienced the brunt of these events. And although it can't be explicitly said, signs of these algae blooms occurring may indicate some excess nutrients or eutrophication in the system. We began sampling Coral Creek in July of 2014 and ended our directed sampling project in June of 2022, so 8 years. We used a fisheries independent monitoring stratified random sampling design, and we broke up Coral Creek into 3 distinct sections, or creeks, so east, west, and lower, and that's our study creek. And then we encompassed these 2 characteristically similar and adjacent creeks, Catfish and Whidden, which are also larger, shallow, and associated with seagrass in the systems, but have markedly lower development, and right along their shorelines there is our reference creeks. We sampled each creek monthly with a 21-meter seine that we set from the back of a boat, where we enumerated and identified each fish to the lowest practical taxonomic level, and then characterized the habitat and water conditions at the time of sampling in our sampling area. Biologists working in the system over the years kind of began noting that there seemed to be some apparent dips in seagrass coverage within these creeks. And when we began looking at these data, sure enough, that's what we saw. And if I can draw your attention to that red line there, that's the eastern portion of Coral Creek. You can see it going from almost 80% coverage in our samples, flatlining to almost 0 at the end of the study period. In comparison, Whidden and Catfish, our reference creeks, kind of hover from 20 to 40 peaks and valleys there, and are actually trending upwards at the end of our study period. So because of what we saw moving forward, we kind of turned this, what we see in East Coral Creek, as a seagrass die-off, and assigned it seagrass die-off stages. So years 1 to 2 were pre, years 3 to 5 were transition, and years 6 to 8 were post. And this led us to some study questions of, first off, how do fish communities differ between these creeks and seagrass die-off stages? And then, is there a relationship between fish community structure and the seagrass coverage in these creeks? Uh-oh, my plot is missing. Everybody's biggest fear in a presentation. Well, that should be a Permanova table there showing 3 different terms, one being the difference between creeks, one being the seagrass die-off stages, and then the interaction before these two terms. And what we found was there was, and I guess you'll have to take my word for it, a significant effect of creek and seagrass die-off stage. So the fish communities differed significantly between creeks as well as those die-off stages. But most importantly, and that's still there in red luckily, we found an interaction between these 2 things. And pairwise testing confirmed that the effective seagrass die-off stage differed between creek systems. So for instance, in one of our reference creeks, those pre-transition and post stages, there was no significant difference between those fish communities, whereas in one of our study creeks, such as East Coral Creek, there was. Now, I'm really going to hope this other plot shows up. Alrighty, so for the second question, we turn towards a constrained multivariate analysis technique called a canonical analysis of principal coordinates, or CAP for short. And I'm going to kind of walk you through this plot because these multivariate plots are a little dense, but each point here on the graph represents the average fish community structure for a given year in a given creek. Now this might look a little different to some of us that are familiar with this technique because we used a single axis here that explains about 41% of the variation in the data cloud, and then oriented that with the corresponding seagrass cover in a creek for a given year. So if you look along that x-axis, the points along that x-axis, if they're closer together, they are more similar. If they're further apart, they're dissimilar. And long story short, there is a substantial relationship between seagrass coverage and fish community structure. And that's exemplified there by that canonical coefficient up in the corner there at 0.83, which is analogous to an R-squared coefficient. So if we apply like numeric labels showing which point is from which sample year, you really see the story kind of come together. Whew, this showed up, that's good. So you can see in Eastern Coral Creek, if we just look at East Coral Creek and Catfish, for example, Catfish Creek kind of bounces around. There's no real temporal trend there. However, in East Coral Creek, you can really see that trend come out, almost a monotonic shift in structure that's related to the seagrass cover over the sample period. And then again, here are those groups that pre-transition and post, and I'd just like to remind you guys that they were in fact statistically significant here in East Coral Creek. So if we look at some of the species driving the differences between this pre and post group in East Coral Creek, here on the x-axis is mean abundance. The black bars are going to be in the pre-group, and the gray bars are going to be in the post-group. And you can see that there's these large abundances. For instance, that top species there, L. parva, that's the rainwater killifish, so the first fish pictured on the left of this graph here, have these large abundances in the pre-group that are then lost in the post-group. Similar with other things like the sailfin molly and the pinfish are being offset by these increases in abundance in the post-group of these kind of more so habitat generalist and planktivorous species such as the bay anchovy, the tidewater mohara, and the striped mohara. So we see this significant trend over time, this change over time that's related to seagrass loss or declines. And it's hard not to think about this idea of a phase shift. So just to orient us with what a phase shift might be, we can have this classic rolling ball model where you can think of that green ball there as kind of the community itself. And on the left of this slide, in that phase one, that would be the healthy state for, say, this estuary, this area, abundant seagrass, clear water, marsh and seagrass-associated fishes. And as it's in that kind of basin there, it's dynamic, right? There's stressors, there's things acting on it to move, things are changing, it's far from static. You have increases in seagrass in abundance, dips, changes in abundance of certain fish species. But over time, if stressors such as increased nutrients intensify or persist, you might have that right side of the basin there kind of dip down and get worn down. And you can think of that right side of that basin as kind of being the resilience in the community. So as these stressors wear down the resilience, it gets a bit easier for this ball to reach that red dotted line or that tipping point or threshold and kind of roll into maybe a new phase. So we believe this may be what we're seeing in fish, is maybe a signature of this. So it's just something important to kind of bear in mind. Now, what are some implications of a shift such as this in Coral Creek? And just a few things, and all of this, again, requires future research to really pin down, but restructuring of the food webs, altered resilience in response to other disturbances such as red tide events, changes in productivity, and again, really need more research to kind of pin it down there. So I mentioned earlier that Coral Creek is characteristically unique, and it's important to point out that we have lots of tidal systems in Charlotte Harbor that are not associated with seagrass that seem to function very well, right? So where does this really fit into the bigger picture of things? Where are we, what we're seeing here? And that figure on the left there, you can see it. If I can draw your attention to the eastern portion of Charlotte Harbor here for a minute, you can see these losses in seagrass from 2018 to 2020, coupled with this increased persistent, that figure on the right, this increased persistence and abundance of filamentous green algae. So what we're seeing in Coral Creek on that smaller scale in an area that may show effects sooner than on an estuary-wide scale just because it's smaller is what we're seeing kind of a warning sign of what could be estuary-wide change. Now, although that seems very doom and gloom, I'd like to think that a warning sign is really a call to action to be able to do something. Now we know what's going on. We have a finger on the pulse. We can, take action here. So moving forward, one thing that popped out to us was scale is very important. So in terms of Coral Creek itself, a smaller system which may be kind of closer to that tipping point or even rolling down that edge, a restoration focus here along with conservation, preservation, and mitigation actions are going to be really important. And I have a restoration asterisk there because these efforts are actually currently ongoing. And so continuing to identify drivers of seagrass loss goes hand in hand with continued monitoring and research there, as well as stakeholder cooperation, and I should have added a word collaboration as well. And if we zoom out to kind of more so an estuarine scale, where we're probably not necessarily there just yet, but again, it's easier, especially on a larger scale, to prevent these changes from happening than it is to try to fix them if they do. So conservation, preservation, and mitigation focus, along with restoration, and then continued monitoring and research as well. And again, that stakeholder collaboration and cooperation is critical. So in summary, these are a lot of points that I basically just went over. I'm not going to touch on every single one, but I would like to take the time here to really draw your attention to that bottom one there about stakeholder cooperation. And if you look around this room, you'll see lots of folks from different areas. And when I mean stakeholder, I mean your state agencies, NGOs, academic institutions, the public. It takes a village to address these issues, especially on a larger scale. And if you look around this room, you can certainly see that in action here. So with that, I'd like to say thank you, especially to all the folks at the Charlotte Harbor Lab who helped compile this robust data set, as well as Charlotte County Restore for providing some of the funding to conduct this research. And I look forward to taking any questions at the end of the session. Thanks.
Hello. All right. So sticking with the aquatic habitat theme so far for this session, we're going to be looking at the habitat utilized by juvenile tarpon when they first recruit to the Charlotte Harbor system. So this presentation is titled Juvenile Tarpon Emigration from Ephemerally Connected Coastal Ponds in Southwest Florida. So fish may utilize a range of different nursery habitats during early life stages. But for this presentation, we'll be looking at the habitat that tarpon utilize, which are these ephemerally connected coastal wetland ponds. By ephemerally connected, we mean that these ponds receive only a brief connection throughout the year between the ponds and the adjacent estuary. These ponds are typically located at the termination of tidal creeks. As these tidal creeks reach areas of higher elevation, they become disconnected from the adjacent estuary. It's hypothesized that juvenile tarpon move into these ponds during the wet season, which coincides with their spawning season. And they will then remain within these habitats throughout the dry season, keeping them relatively safe from predation while also giving them access to high densities of prey, such as typical marsh species such as mosquito fish and mollies. What we're then interested in is how exactly are these fish then getting back out after remaining in these ponds for about a year or so and reaching these relatively larger sizes. So we broke the study down into two different objectives to determine how these ponds are functioning as nurseries for these tarpon. Objective 1 was to determine these opportunities for emigration using acoustic telemetry matched with key physical and biological data. And Objective 2 was to determine how the segregation of these isolated ponds was affecting the body condition in these groups of fish. So here's a breakdown of our study site. Our study site is located on the Cape Hayes Peninsula here in Charlotte Harbor. The Cape Hayes Peninsula is a part of the Charlotte Harbor Buffer Preserve State Park, so this area holds relatively untouched coastal wetland habitat. On the right, you can see our study area. You want to pay close attention to the slide here as I'll go through some different terms and designations that will track throughout the rest of the presentation. So our study area is broken down into two different connection types. We have an anthropogenically altered ditch connection, which was created from derelict mosquito ditches created in the 50s and 60s. And then we also have a natural marsh connection, which consists of salt marsh vegetation, salt flats, and mangrove forest. We further broke down each one of these connection types into different pond designations based off of this pond's location in relation to the adjacent estuary. So the pond closest to the creek system, we have the proximal pond, then the mesial pond, and the pond set furthest away from the creek is the distal pond. For tagging, we used a 40-meter seine deployed from a kayak, and the tarpon that we tagged during the study ranged from 330 to 600 millimeters total length, so we were tagging fish that are about 12 to 15 inches long. We tag these fish using Vemco V9 acoustic tags, and this allows us to track the movement of these fish within our array for about a year and a half until the battery life of these tags were reached. So here's kind of a breakdown of what acoustic telemetry is and what our acoustic array looks like. So these acoustic tags emit a series of pings, which are decoded by these acoustic receivers. These acoustic receivers store these individual tag IDs based off the date and time that it picked up this tag. And so our hopes were to detect these fish as they move from pond to the adjacent creek system, showing successful emigration from pond to the estuary. And so what we wanted to also figure out was what may be prompting the shift from pond to creek system. So we collected data related to suspected cues for emigration drivers. These consisted of water level data, age and growth, pond connection type and distance, barometric pressure, and water temperature, and we tested these using mixed effect logistic regression models. So what we did is we tested all data of the fish being present within the pond against what was occurring at the time of emigration to see if there was anything significant that occurred at the time of emigration. And what do we learn from these models? We learned that it's a culmination of factors occurring simultaneously which prompt this habitat shift from coastal wetland pond to estuary. We saw huge spikes in water levels on the day of emigration for these fish. We saw low barometric pressure was a driving factor as well, which could be a proxy for a possible storm event occurring at the time of emigration. That estimated fish size was a factor that these fish may have to reach potential size until they make the shift from pond to creek system. A drop in water temperature, which could be caused by either a rain event or storm surge. And we also saw that the pond distance from connecting creek and connection type also played a role. So now we know kind of what ideas, or what may be prompting these movements from creek system or from pond to creek system. But let's look at some of the actual events which transpired to allow for these fish to emigrate from pond to creek system. So we have an abacus plot showing our 2019-2020 tagging data. Across the x-axis is the date, and y-axis is each individually tagged fish. Every time you see a color change here going across the x-axis is the fish changing position to a new position in the nursery. So you can see at the top of the plot, all those fish remain in purple that were tagged in the distal marsh for the entirety of that tagging year. Or in the bottom left, outlined in black, you can see those fish begin as green in March and then all transition to blue, showing successful emigration from the pond to the creek system in August of 2019. Which made us wonder what occurred in August 2019 to allow for all these fish to move from the pond to the creek system. So in August, we saw a large spike in water level caused by a high tide in the creek system. And this allowed for the water to move through the creek system, flood the marsh, and bridge the gap between the pond and the creek system, allowing for these fish to emigrate from the pond to the creek system. 2020, we resumed tagging, and as you can see from this plot, there's a lot more colors on this plot. So remember, every time you see a color change across the x-axis is a fish transitioning to a new position in the nursery. So in 2020, we saw fish emigrate from all ponds that we had fish tagged in that year, but what was so different in 2020 compared to years prior? So 2020 was a highly active hurricane season, producing the most named storms on record in the Atlantic Ocean, and these large tropical cyclones is what gave these fish the opportunity to move from pond to creek system. So here we have a couple of events which allowed for emigration in 2020. On the right is water level data, and on the left, again, is our abacus plot with a couple of those storm events outlined. So during Tropical Storm Cristobal, we saw fish emigrate from the proximal pond. During Tropical Storm Sally, we saw fish emigrate again from the proximal pond as well as in the mesial pond. And then in 2020, we also saw our first emigration of fish from the distal pond, the ponds that furthest back in the landscape that requires these kind of massive flooding events to allow for emigration. And we saw that during the events caused by Hurricane Eta. So now we'll kind of look at like one individual fish's story that emigrated during the events caused by Hurricane Eta from the distal marsh pond. So Hurricane Eta approached Florida's coastline and made landfall north of Charlotte Harbor on November 11th at around 7 p.m. So if you look at that white box outlined in red, it will show the progression of time as this fish begins to emigrate and Hurricane Eta approaches Florida and storm surge begins. So currently, the fish moves from the distal marsh pond into the mesial marsh pond on November 11th at 5:54 p.m. Then by 11:38 p.m., this fish successfully emigrates from the pond into the tidal creek. So what this fish just did was, in a little under 6 hours, traveled 1.3 kilometers through dense mangroves filled with pneumatophores, prop roots, fallen trees, over salt flats, and through salt marsh vegetation, all to reach the headwaters of the tidal creek before the floodwaters recede and leaving the fish stranded on the marsh. So what do we learn from our telemetry data? We learned that emigration is linked to high water events, and we saw fish emigrate during 10 distinct high water events. During the study, 44% of the fish that we tagged emigrated during tropical cyclones. We were also able to gauge relative survivability of fish in these habitats. So 27 of the 53 fish that we tagged were detected emigrating, and 19 more of these fish, although they didn't emigrate, we were able to track them past 1 year until the battery life was reached within these habitats. Although the fates for these fish were ultimately unknown, we were able to track these fish for at least a year, showing them able to reach relatively larger sizes, which gave them a much greater chance of survival. And this indicates an 87% apparent survival of tarpon in these habitats. Another question that we had was, how does this kind of isolated, segregated pond system affect the body condition of fish in these habitats? With telemetry, we learned that these fish kind of weren't mingling and going back and forth between the proximal, mesial, or distal pond, or possibly going from pond system to creek system. That these fish were remaining in the pond until they had an opportunity to move from the pond to creek system. So we want to know, is there variation in body condition among each one of these ponds, possibly from things such as increased predation risk or possibly high densities of fish causing more competition for prey sources. So visually, all these fish in these habitats appeared healthy. They all seemed comparable, but we wanted a way to kind of analyze these different habitats to assess for variation in body condition. So we used a generalized linear model to compare the standard length of fish in relation to the dorsal height. And what we see with the plot on the right was that all ponds had comparable body condition. That whether a fish was located in the proximal, mesial, or distal location, they all had comparable body condition or fitness in each one of these habitats. We also used scales to determine different age cohorts in each one of these habitats. These scales gave us a non-lethal way to determine between whether a fish was young of the year or age 0 or not yet 1 year old, or if the fish was 1 year old or greater. What we determined was that there's a 300 millimeter threshold to represent 1 year old fish or greater. So fish that are 300 millimeters or less are young of the year fish. Fish that are 300 millimeters or greater are age 1 or older. And just for reference, these fish are about 15 inches total length. So now we'll transpose that kind of 300 millimeter threshold onto each one of these length frequency plots. Here we're looking at the proximal marsh pond. So here you can see that gray line is the 300 millimeter mark indicating age 1 or greater fish. So all fish to the right of the gray line are at least 1 year old or greater. All fish to the left are young of the year fish. And what you want to make note on for this slide here is that all 3 years, 2019, 2020, and 2021, this proximal marsh pond mostly consists of young of the year fish. Typically, this could be assumed that this kind of lack of older, larger fish could be due to something such as mortality from predation or possibly poor habitat causing slow growth in these habitats. But from our telemetry data, we know that these fish are actually experiencing annual emigration from these habitats, and that this lack of older fish is actually due to fish coming in and leaving the following year consistently. Compared to habitats such as the distal marsh pond, again, we have the same gray line indicating all fish to the left being young of the year, fish to the right being age 1 here in the distal marsh pond. And what you want to take note on in 2019 to 2020 is the much greater presence of age 1 fish in these habitats. Then if you look at 2021 and compare to those 2 years prior, you see a higher grouping of young of the year fish to the left of that gray line, and you see a reduction of age 1 plus fish. As you recall from our telemetry data, 2020 was a highly active hurricane season, and this allowed for ample opportunities for emigration from this distal marsh pond. So in 2021, we see a reduction of those age 1 fish because they were able to emigrate the year prior, and we also see an increase in young of the year fish, showing that these large tropical cyclones act as not only an opportunity for emigration from these habitats, but also an opportunity for fish to recruit into these habitats. We also learned that these habitats are not functioning as population sinks, that these tarpon are capable of surviving in these habitats for multiple years until they have the correct opportunity to emigrate. So to wrap up kind of this whole study, what all did we learn? We learned that high water events are required for emigration, whether it's caused by tidal events, rainfall, or large tropical cyclone events causing storm surge in the area. We learned that body condition was comparable among all the ponds, that whether a fish was located in the proximal, mesial, or distal pond, the body condition was suitable in all habitats. And we also learned that flooding frequency affects emigration opportunities, that some fish may require or may have annual emigration opportunities, or some fish may remain in ponds for multiple years until their opportunity for emigration arises. We were able to document the contribution of juvenile tarpon to the estuarine population. Although we weren't able to track these fish all the way to adulthood and maturity, we were able to track these fish to relatively larger sizes, which gave them a much greater chance of survival once they reach this estuary, which ultimately, in hopes, they contribute to the adult population of tarpon. And we also gained knowledge which can be utilized in future restoration projects, things like how these habitats function as tarpon nurseries, like the flooding frequency and the extent of the flooding which is required for these tarpon habitats. So in order to support a healthy fishery, it starts with healthy nurseries. The tarpon fishery is unique as anglers target these fish anywhere from 6 inches to 6 feet long and everything in between. And all this can be found right here in Charlotte Harbor. So in order to protect this fishery for years to come, it begins with protecting these nursery habitats. And this means not just the nursery habitats themselves, but also the surrounding areas, from things such as development, causing changes in hydrological connections, as well as increased nutrients. So here we'll look at kind of one small scenario that's playing out here in Southwest Florida, but has played out all throughout coastal Florida and is ongoing today. So here in the bottom left, you see these ponds that we just discussed as being highly productive tarpon nursery sites. You see the proximal, mesial, and distal marsh habitats. And on the right, you can see the proposed zoning to take place around these habitats. Things like low density and medium density residential, as well as commercial zoning. Although these habitats themselves are protected from development, they're still very susceptible to changes in hydrological connectivity due to them kind of lying on the land-water interface between these areas. There's also the opportunity for increased nutrients into these areas that we learned from previous presentations, which can cause issues to these systems as well. So in order to protect this fishery for years to come, kind of an active approach must be taken to document and protect these highly productive nursery areas to protect the fishery for years to come. And so with that, I'd like to thank everybody at the Charlotte Harbor Field Lab and all my funding sources. And I guess we're set for questions.
Well, now we're going to go into our question and answer, and I noticed that some of you have been able to join us over the course of this first panel, so I thought I would take a moment just to kind of refresh on what we're going to do here with regards to Q&A. We're using a tool called Minty. If you need Wi-Fi, you're going to look for the Wi-Fi network that simply says vendor, and then the password to that Wi-Fi is 75Taylor, with the T capitalized, capital Taylor. So if you need Wi-Fi, feel free to log in to your Wi-Fi and go ahead and open up minty.com. So we already have some questions coming in. You can see there at the very top, the code is 8217994. So go ahead, if you haven't already, enter your questions now, and I will turn it over to our panel for you all to self-select questions and take turns picking a question, answering it. Maybe some of your fellow colleagues might want to chime in answering that question or adding to your response, but go ahead and do as many as time allows. We'll cue you when you get close to your Q&A time, but we've allowed abundant Q&A time in each of our panels to really allow for, a substantial audience interaction. So go ahead. Thank you. Want to go down in row, just pick. So if you could state the question and then just give your response. If there's a question up there that you see that you are interested in responding to, just go ahead and state the question and then start responding.
Okay. How do we scroll or they?
They're scrolling, but maybe we'll keep it slow. We'll just keep on the top for a minute and then we'll scroll down after you've had a chance to answer a couple at the top. But I know we have a lot. We already have 44 questions in for you.
All right, so the first question that I see is how many adult female sawfish are there in your study area that are known pup-producing size? I think it's safe to say that we don't really know the actual answer to that, but there was a study done, I believe in 2014, looking at the fin clips from all the juveniles that we had taken in our sampling, and looking at the genetics from those and translating that back to how many females were producing pups during that time period. And I believe in Charlotte Harbor, there was found to be 55 females that were contributing to the population of Charlotte Harbor. At any one time, perhaps 10 or 12 for each nursery within Charlotte Harbor.
Okay, the question is, what is a ball bungee and what is its purpose? So a ball bungee are the bands found that connect the boat lift cover to the boat lift itself. So the band and strap together in conjunction is what holds your boat lift canopy on your cover. They're used in other various things of canopy-like. You'll see a lot of ball bungees on those as well, but mainly for these purposes, because boat lifts are on the water and in the canals, it's more specific ball bungees to those boat lift covers. So I can also quickly answer, are you seeing any entanglements with things other than fishing gear or elastics? What about products labeled biodegradable? To be honest, we haven't come across any products that are labeled biodegradable. Other than elastic bands, yes, we've had various other forms. Kind of like I've mentioned, there were sun visors, plastic bags, a dog toy, onion bag, pretty much anything that can become entwined on a very toothy appendage can very easily get wrapped up. And unfortunately, as Andrew kind of mentioned in his, within their first year, they double in size. So certain objects of marine debris can cause really severe consequences on the fish since it doubles within that first year. And hopefully that answered that question.
All right, so I see a question. Tidal Creek habitat change and baseline species biodiversity decline seems to be affecting our juvenile game fish nursery habitat. What is your data showing? To kind of summarize it, what these data show that I presented today is that there has been a shift in the fish communities we sampled within that creek over time, and it's related to that change in seagrass within the creeks that we were studying. So it probably is important to point out that when we sample with these small nets, there's a little bit of gear selectivity going on. So we're talking about more so small fish or juvenile species, or juvenile fish of like game fish species like trout and redfish, stuff like that. So that community there might be a little different from if you're thinking of, maybe larger conspecifics like snook, redfish. So basically, it's showing that there's been a significant change in the assemblage structure over time related to seagrass loss. And I hope that kind of answered the question. If not, please feel free to come grab me later.
All right, so I see one here for me is, do you study elsewhere in the county? Can we submit info on other areas? We have witnessed tarpon movement on rain flood tides from varial estuarine ponds. Currently, we are not studying elsewhere in the county. We are kind of in the works of branching out to kind of the eastern side of Charlotte Harbor, so that's an area we potentially might kind of mimic this study that we did there on the Cape Hayes Peninsula over on that side of the Charlotte Harbor. And can we submit info on other areas we have witnessed tarpon movement? Yes, definitely. I'm always interested to kind of look into other areas and see how other areas may be functioning in comparison to this. I know other agencies are also trying to kind of take into account these juvenile tarpon habitats throughout the state and trying to build a record of these areas. So yes, that's definitely something we're interested in as well.
I noticed a question, I think we scrolled past it, that's okay. It was about if sawfish are still using our bigger river systems in Charlotte Harbor and whether or not they can still be found up near, oh, there it is, up near the freshwater influx. To answer that question, yes, we do still catch sawfish in our sampling, as well as get reports of sawfish relatively far up in the Peace River and the Caloosahatchee River, and somewhat in the Myakka as well. But we have caught sawfish in purely freshwater in both river systems, and in the Caloosahatchee specifically, we've caught and seen sawfish all the way up as far as the Franklin Locks. So that's about as far as you could possibly expect to see a sawfish.
See another one here too while we're here for a second. It says, having grown up fishing, somebody who knows me, our estuary and tidal creeks in the harbor and sound, this drastic change in baseline species and phytoplankton accumulation has deteriorated water quality. Any hope? As I am more of a fishery scientist and do not specifically study water quality, and kind of the data we capture in our sampling is more so the conditions, so salinity, temperature, pH, stuff like that at the time of sampling. I can't speak too much directly to the water quality stuff, but as far as the any hope, I feel like that's a really important point to touch on, is that I do think there's hope, right? I mean, again, if you look around this room, we're all here for a similar purpose, and that's to address issues like this and come together, collaborate, and try to kind of turn the tide on them. So I do believe that although Coral Creek specifically might be showing these changes, you know, we can do something about this and hopefully reverse it. So yes. Short answer, yes, definitely some hope there.
All right, so Dylan wants me to answer this question. How are gill nets used for sawfish sampling if they are banned? Fortunately, we're able to get permission from the federal government to use gill nets in our sampling, and we actually set these gill nets for only an hour maximum. We check them every half hour, or if something is found to be entangled in the net, so if we see anything splashing around, we check them immediately. And sawfish are known to be a very tough species. They handle stress very well, so as long as we're actively checking them and we're handling these sawfish with care, there's not much of a concern for the sawfish themselves, and we do have federal permits that allow us to do this, even though they're banned for recreational and commercial use in the state of Florida.
I see one talking about creek fish assemblages there. I'll take a whack at. Regarding creek fish assemblages, the data for this study were all collected after Hurricane Charlie. Any idea what the assemblages looked like before Hurricane Charlie? To my knowledge, and there may be some other people in the room that can answer this because that was long before my time here with FWC, we probably do have some samples that fell out randomly in our universe from Coral Creek and those creeks, so we might be able to look at that. But short answer is no. I'm not really sure what it looked like pre-Charlie, but I just wanted to address that one since I know hurricanes are probably on the forefront of everybody's mind.
If you wouldn't mind scrolling up, I can answer one. Yeah, look, right there. So what is the function of the rostrum on small tooth sawfish? Can you find them towards the shore or offshore? What are some differences? With gill and hook nets, what does STL stand for? So I'll start with that one. That stands for stretch total length of a fish, one of our basic measurements. The function of the rostrum is mainly used for feeding and defense. And yes, you could see throughout Andrew's presentation, you can find fish inshore and offshore, just depending on size class and those shifts. And then some differences with gill and hook nets. Typically, hook nets have some sort of bait on it to attract and try to lure our targeted species in. Gill nets do not have any bait associated with those.
There's a question there that says, was the larger outlier female in the shallow water perhaps there to pup, and when is its pupping season? It's possible. It is definitely possible. We're not entirely sure. We'd have to look more at the genetics from the juveniles we also captured that year. We don't believe, based on some preliminary genetics looking, that it was there to pup, but it was there in the right season. Their pupping usually takes place here in Charlotte Harbor between kind of, I'd say, February through early summer. So we caught that fish in June, I think, and that would make sense. It would be possible.
Scroll down a little bit more.
All right, there's a question there. It says, regarding sawfish habitat, what are the effects of salinity changes associated with freshwater flows? These flows are changing due to human impacts, so there are management implications. There was a study done a few years ago looking at the effects of salinity on sawfish movements throughout the rivers here in Charlotte Harbor. And we found that there are impacts, or there are effects of salinity changes on sawfish, and that's mainly on the juveniles. As I mentioned, for the most part, the juveniles are the ones spending the majority of their time in the rivers, so they're more likely to be affected by salinity. And we found that there's kind of a lag where once those freshwater outflows start coming down the rivers, the juvenile sawfish will still be spending time up in the freshwater, and then as that freshwater outflow continues, there's a couple month lag where they'll move down into the lower parts of the rivers. And even, for example, just yesterday, we caught a sawfish in almost purely freshwater up near downtown Fort Myers. So they do spend time in freshwater, but that's generally the small juveniles.
I see two questions here that I think I can kind of knock out in one stroke. The first one being, is this connection between grass and fish ecology applicable in other areas of the harbor? Meaning, are there other seagrass-associated creeks that function similarly? We do have, as far as knowing how they function, we do have other creeks that do have that seagrass association within the harbor, such as Catfish and Whidden are two reference creeks. And they do appear to have, to function, those two do seem to function very similar to each other now, and in this study, they look a lot different than Coral Creek, but that might change and say we had a data set that looked way back in time before all that habitat alteration within Coral Creek. Then the second question is, how do the results from this study of creek fish communities compare to other tidal creeks? Note that prior to Hurricane Charlie, most of the creeks on the east wall, for example, had seagrass. And that is a great question and something that I would love to look into, if we have data there to look at that, because that is definitely true. There were those east wall creeks, especially towards the upper section, that did have seagrass association with them as well. So it's a pretty good point there.
I could take a couple that are up there right now. One question is, the juvenile tarpon from these remote ponds I have noticed to have interesting physiology, such as a disproportionately large tail. Is it stunting or evolution to be the fastest to the food source? In the system that we studied, we didn't see any kind of disproportionately sized fish. All the fish seemed to appear to be healthy, similar body condition among all the ponds studied. That being said, I mean, we studied a large amount of ponds on the Cape Hayes Peninsula, but there could be some other scenarios playing out in other areas that we haven't noticed. So personally, no, we haven't seen any of these kind of variation within the ponds. Another question, have you looked at moon phase paired with tides and how that affects ability of fish to emigrate from ponds to creeks? Not at moon phases directly. It's something I did consider was kind of lunar phases, how it may have an influence on fish movement. This kind of goes hand in hand, like you said, with high tide, full moons. So there definitely is a potential. There are a lot of subtle nuances that go on with the kind of the immigration opportunities that allow for these fish, that it may be a high tide, but also a high tide which may coincide with a particular wind direction to kind of amplify that high tide just a little bit more to kind of bridge that gap just a little bit more so that these fish can't get out of there. Another one, how did you access the ponds for sampling? A lot of hard work. A lot of these ponds were extremely remote, kind of no one previously accessed them, so we kind of blazed our own trails getting into these habitats. A lot of Google Earth satellite imagery, walking around in circles out in the marsh, figuring out how the heck to get into these ponds. So a lot of hard work, a lot of mosquitoes.
I think all of us can speak to that because we helped out a little bit there. Hoofing that big net around through a mangrove forest, if you've ever seen prop roots and how that looks, is definitely tricky. I also wanted to address a question here. It says, what species was in Coral Creek? Rupia, lost possibly due to change in salinity. So I'm glad someone kind of pointed out the species component of the seagrass side of things, but I am not a seagrass ecologist. I can tell you, however, that in East Coral Creek in those beginning years where that really high abundance was, there was a lot of rupia in that system. However, in Coral Creek and catfish, for instance, now we mostly see a lot of haliduli, similar with down in the other portions of Coral Creek. So although I can't really answer was it due to salinity changes, I do look forward to collaborating and speaking with some seagrass ecologists and kind of getting their input on that for sure, because that's definitely an interesting story there.
I got a couple here. So as sea level continues to rise, do you expect an increase in tarpon emigration to tidal creeks? Definitely as sea level continues to rise, I mean, the opportunities for fish to move into these creeks and into the ponds does create more potential. But sea level rise may actually negatively affect these habitats as well by kind of increasingly giving too much of an opportunity for these ponds to become flooded, giving access to these habitats from larger predatory fish like snook or jacks, kind of defeating the nursery purpose of these habitats. Or kind of previously, with the natural movement of sea level rise, these ponds would kind of just move further and further back in the landscape. But now the issue with many of these coastal wetland ponds is that with them landing on the, or being located on the land-water interface, the kind of natural progression of these habitats is now blocked by urban development. So kind of a lot of these habitats are going to soon be squeezed out from this from the seawater rise. And what effects will storm surge have on these remote tarpon nurseries now that the water is still foul from storm surge pollution? That is a good question. As far as storm surge pollution, from what we've seen, these tarpon are able to tolerate some pretty harsh water quality conditions, going from anoxic to water temps in the 90s. They're able to survive in them, so they're able to tolerate some pretty harsh conditions. I don't know if that really gets to your question. Do we know if the fish are using other ditch canal systems not connected to ponds, such as Wolverton? I'm not familiar with where Wolverton is, but growing up in Florida, we would see kind of tarpon all throughout kind of urban areas and stormwater ponds, canal systems, golf courses all over. And yes, they definitely use these habitats. That's something that I'm personally interested in, is kind of how these urbanized residential areas are functioning as potential nursery habitats, or are these fish kind of getting into these areas through small culverts, and are they trapped in these areas, or are they still able to get back out? So is this something that we could possibly improve upon with different stormwater designs to allow for kind of changes in floodgate designs to allow for kind of inward and outward movement of these fish? Are there other high water events that will facilitate tarpon emigration? Will you look at the area to see how Hurricane Ian was impactful? Yeah, there's definitely a range of different events which allow for emigration. As far as Hurricane Ian, we did have fish tagged during this study. It was kind of interesting during Hurricane Ian. So we had fish tagged in the Messiel Pond that kind of just carried over from the previous year. We actually didn't tag this past year, so we didn't really get the full picture of what happened during Hurricane Ian. We saw fish go missing from our acoustic array within the pond system following the effects from Hurricane Ian. Up in this area, we didn't see any storm surge for Hurricane Ian. We largely saw the rain events. We mainly saw effects from the large amount of rain event. And potentially what we think might have happened during Hurricane Ian was the fish attempted to emigrate, and either they kind of just skipped over our acoustic array and we never detected them, or they potentially had some type of mortality during emigration because we saw them go missing from our pond system but never were detected in the creek system. That's kind of an easy one. Do you anticipate more recruitment due to Hurricane Ian? Yes, I do, because All throughout, following 2020, just for an example, once we had that highly active tropical storm season with the hurricanes, we saw massive recruitment, not just here in Charlotte Harbor, but also another study site that we have up in Manatee County, up in Robinson Preserve. We saw tarpon up in that habitat and pretty dense populations as well. So yes, I think there probably will be kind of high densities of tarpon sprinkled out throughout ponds that may have previously not had fish for the past couple of years. I got one. What stormwater ponds are you suggesting? How do you envision it happening? So in order to make most of Florida livable for people, we have to kind of drain a lot of this land in order to take the water off of the habitats. So in order to do this, we kind of built up a bunch of our developed areas, and there's stormwater ponds kind of sprinkled all throughout these coastal systems. So kind of something I'm envisioning is kind of turning these stormwater ponds into potential nursery sites, which could be done pretty simple with, I mean, a little bit of engineering, which is not my forte, but just basically making stormwater friendly floodgates that allow for this inward and outward movement of fish.
I noticed one question up above asking about what type of restoration efforts are occurring on Coral Creek. And to my knowledge, there are currently hydrological restoration projects underway spearheaded by the Southwest Florida Water Management District. I know this is I believe a multi-agency or a collaborative effort. I'm not exactly sure who all is involved, but to my knowledge, it's hydrologic restoration. And there are probably other people in this room, again, who can speak a little bit more on this, so I apologize if I got that wrong in any way, but I did just want to address that question since I pointed it out in my talk.
I got one. How large are the juveniles when they travel into the ponds? This is a great question. So tarpon, as juveniles, they undergo a leptocephalus larval stage, which is essentially a translucent kind of eel-like stage. We are not exactly sure what exactly the size is as they move into these habitats. We've been sampling kind of Charlotte Harbor for the past 30 years, and we've never caught a leptocephalus tarpon. We've caught other things. We've caught ladyfish. We've caught eels. We've never caught a tarpon. In this habitat, we have caught post-lepto size fish that are about, I think the smallest one was around 20 millimeters. So we really don't know exactly. Are they coming in as leptocephalus and then going through metamorphosis into kind of the post-lepto stage once they reach the ponds? Or is this something they do once they reach the estuary and then swimming into them as kind of post-leptos? How important are tarpon to the environment? I mean, I think every species kind of serves its role in the environment. But I mean, let's just say the environment here in Southwest Florida in particular, it's a huge economic driver in this area. It's a billion-dollar industry with the tarpon fishing capital of the world being located right here in Boca Grande. How do the juvenile tarpon get into the backwater areas? Again, this is one of the things we're not really exactly sure. I envision it's kind of some type of cue that they're picking up on with the fresh water or possibly detritus that is located in there, so they're able to kind of sniff it out essentially as they're moving into the estuary, moving into these areas. Yeah.
There's one there that says sawfish. Do you see an opportunity to help fish by adding structures for cover for fish that are transitioning to deeper water? And what might those structures look like? I don't necessarily think that would be something that we would need to do for sawfish because, as I mentioned, they grow extremely quickly in their first couple of years. So they're doubling in size to about 6 feet long in their first year or two. And then by age 2, they're over 2 meters long generally. So when these fish start to transition into deeper waters, as long as they're staying in the rivers, which is typically what we're seeing, I don't believe there's all that much risk for a 6, 7, 8, maybe even 10-foot sawfish in regards to predation while they're in the river system. We do have bull sharks in the Caloosahatchee and the Peace Rivers, but generally those are also juvenile bull sharks. So I think they can usually fend for themselves at the sizes where they're going to start to actually transition. That's why those small juveniles are spending their time in very, very shallow water, trying to evade predators and get a little bit larger before they switch into deep water.
See a question there. How does the shift from SAV to macroalgae affect the swine or gear? I noticed more gobies, smaller, and fewer pinkish, pink shrimp? I'm not sure what that is. But I think it is important to kind of point out that when we do have more of this macroalgae in the system, it can affect a little bit of how that gear fishes. So for instance, if we have massive amounts of green algae we're setting in, we might be, when we go to trip the bag and sample, we might be losing some of the fish that are being caught out of there. They could be getting caught in the algae. We try as hard as we can to pick out every single fish that we can, but there could be a little bit of an effect there for sure.
Larval tarpon recruit into proximal mesial and distal ponds. Is the focus on distal ponds due to greater survivability, or does this environment provide a vital life cycle requirement or reduce predation? Yes, as there's kind of a trade-off as these fish push further and further into the landscape. The further they get away from the estuary, the lesser the risk of predation. So like in the proximal pond, for example, we would see kind of higher percentages of snook, and sometimes larger snook, which could potentially predate upon these juvenile tarpon. Where in the distal ponds, these ponds, they're incredibly dynamic, going from, like I said, anoxic to high water temperatures. So pretty much the only large predatory fish that could survive in these habitats were tarpon. So they basically had a free-for-all for all the prey sources they wanted in these habitats. So there's a trade-off. The further they get away from the estuary, they might have as much of an opportunity to emigrate. But then again, they have kind of no opportunities or very small opportunities to be predated upon once they reach these habitats. I got an easy one I get off on. Why high water levels are required for emigration. Basically, without high water levels, these fish aren't able to emigrate. So pretty much the perimeter of these ponds essentially go dry throughout the dry season and even into the wet season. Kind of water will go higher, but not high enough for these fish actually to emigrate across the salt marsh through the mangroves to reach these habitats. So we need these kind of insane high water levels caused by high tides or storm surges in order to even just give these fish the actual opportunity to move. And without that, they kind of are trapped within these habitats for the remaining of the year.
There's a question down there on the bottom left. Do sawfish and nurseries outside of Charlotte Harbor seem to have similar ontogenetic shifts in habitat use? That's a great question, and that's why I'm choosing to keep my master's thesis specifically with Charlotte Harbor, because I think it's important that we recognize the potential differences between different nurseries because of their geophysical differences, geochemical differences. There's things that could be causing these ontogenetic habitat shifts to look different in different habitats and different nurseries. So I'm just focusing on Charlotte Harbor, and I just wanted to address the fact that I don't know if they're different in other places, but because I don't know, I want to leave that open and just talk about Charlotte Harbor for this project, and we'll see if maybe in the future people study similar things in other nurseries.
I got one there. The juvenile tarpon study is interesting. Can you put your findings in context? How many other juvenile tarpon sites are there in Charlotte Harbor? What portion of the population do they contribute? This is a great question. We have gone into, I don't even know at this point how many of these ponds to kind of document the presence of tarpon in the habitats. One thing that we saw is that these habitats are incredibly dynamic, that one year, one particular pond may have a ton of tarpon, next year might have none. So it kind of varies throughout the year. Again, going back to what I was saying before, that certain years may have a recruitment to fish due to maybe a particular west wind, south wind kind of blowing these fish into that particular region of the harbor. And what portion of the population do they contribute? I think just kind of going from what we've been seeing, a high percentage of tarpon in Charlotte Harbor come from these coastal ponds. They will also utilize the tidal creeks in the harbor, and with that, I've seen them mainly in these kind of super tight, narrow tidal creeks. Whether this is the optimal habitat for them, I'm not sure. It's another thing that kind of would require further research and something that I'm interested in is kind of these tidal creeks, so there's lower tier habitat, or are they functioning exactly the same? But I imagine kind of equal populations are equal numbers are contributed from the creeks as well as the ponds. We have not sampled leptos at night, which could be something we could try out with some plankton nets.
All right, I'll take a stab at the bottom middle one on behalf of, I think, all of us sitting up here. But I think this is probably aimed towards sawfish because it says as they grow. It says, does tagging hurt the animals as they grow? What happens if the tags just disappear and stop giving data? So there's a couple ways to answer that. First of all, with sawfish at least, we have documented the recovery of the surgical incisions that we make. So we've recaptured sawfish at different intervals after tagging them, and their ability to heal is incredible. You almost don't even notice after about a week or two. They heal up very quickly. And then after a year at large, after being tagged, you really can't even see the scar. It's pretty amazing. And as far as what happens if the tags just disappear and stop giving data, this is kind of an issue. But this doesn't just happen because of tag malfunctions or fish mortality or anything like that. You have to keep in mind that with acoustic telemetry, the tag is only going to transmit data if it comes into close proximity with an acoustic receiver. So acoustic receiver coverage throughout the state of Florida is very important to make sure that these tags continue to work. We've seen that with sawfish, as they leave Charlotte Harbor, once they leave our acoustic array that I showed on our map, we rely on other people and data sharing networks to pick up our tags in their receiver arrays and other areas around the state and share those data with us. And if there happens to be areas throughout the state where there are no acoustic receivers, then unfortunately we're not going to really hear anything from those sawfish for a period of time until they move into an area where there are acoustic receivers. And we're seeing a little bit of an issue potentially with that down in the Everglades, because the Everglades is just a vast potential habitat for sawfish. There's tons of deep creeks with lots of mangroves, beautiful sawfish habitat, but it's very difficult logistically and permitting-wise to put acoustic receivers in the Everglades. So we might have long gaps of maybe even 2 or 3 years where a sawfish finds an area where there are no acoustic receivers and then comes back into receiver coverage and we hear from it again. So it's not necessarily a bad thing if we stop hearing from them.
There's a question about in the remote ponds for juvenile tarpon, do these habitats help thermoregulate? Darker bottom may keep better temps in winter. Yes, I think there is some form of thermoregulation in these habitats, but mostly that's characterized by kind of a slight depression in a lot of these habitats. And by slight depression, most of these habitats are very shallow, kind of 1 meter or less. And the depression may be kind of an area that's a little bit deeper, like 1.2 meters. And typically we'll see these fish kind of hold out in these types of areas in the ponds.
So quickly, I can answer, were small-tooth sawfish found throughout Florida historically? And the answer is yes. Historically, they ranged from Texas throughout the entire Gulf, all the way around Florida on the East Coast, and up to North Carolina.
Do you suspect that these juvenile tarpon are maturing and staying in Florida's coastal regions, or are they traveling the entire Caribbean Sea in greater distances? I do believe they are probably maturing within Charlotte Harbor, but then joining the adult migratory groups and traveling all throughout the Atlantic.
All right, I think we have time for just one more question. So So if you want to pick one last one.
All right, I think we're going to do for our last one, a question for each panelist. Describe the conservation management applications for your research results. So with the study that I presented, I think the biggest thing is kind of identifying these tarpon nursery habitats so that these areas are known for kind of future developments on what could be disrupted from potential development in the areas. And management applications, again, just kind of keeping these areas in mind whenever areas are proposed for future zoning or development, and also taking this criteria and possibly restoring or creating habitats which have been lost from things such as development.
So to that same question, the conservation and management applications of your research, for the study I presented here, I think the largest takeaway and where this applies is that now we kind of see a warning sign of what could happen on a larger scale potentially. So we know to keep our finger on we know what to look for now. We can start collaborating with other researchers to kind of get to the bottom of these driving forces and really figuring out what can be done to either turn the tide or prevent it from turning altogether. And then the management applications there, I think the same thing, that once we figure out what's really going on, what's driving these changes, if these changes are occurring on a broad scale, that can tie right into management applications to help enact whatever we need to do to kind of turn that tide.
As far as sawfish are concerned and the research that I'm looking at, I think one of the overall reasons that I wanted to look into this is because Charlotte Harbor is one of the designated units of small juvenile critical habitat for the species, the other one being the 10,000 Islands and Everglades. And I think that it's important to look at how these juveniles are using the harbor and the estuary, that whole critical habitat unit, throughout their life, not just as small juveniles, because we could address the fact that this small juvenile critical habitat is also critical habitat for large juveniles and even potentially adults when they come back in to reproduce. And someone also asked a question earlier about the size of the population in Charlotte Harbor, and I think that the research questions that I'm asking could potentially help us to get a handle on how long sawfish are using the harbor before they leave and become migratory, because that would give us an idea of how many different age classes we have in the harbor at any one time, and that would help us to point us in the right direction to see how many sawfish approximately are using the harbor at any one time.
Okay, and for mine, regarding kind of marine debris issues with sawfish, so if we can address and make note of the threats that they still face, we would like to, address those and still figure out what's causing deaths of sawfish. So eventually, it'd be nice if we can talk to manufacturers or stakeholders, but at least, you start with the public and citizens and creating awareness and trying to encourage people to do a little better. So.
Well, join me in thanking all of our panelists this morning. We really appreciate you taking the time to share this important information with us. Thank you.